Rabea Asleh, Mike Gibson coming to you live from ESC 2024. And this is a fascinating topic. How does the gut microbiome affect the phenotypic expression in HFpEF? So fascinating. What did you find?
Thank you, Dr. Gibson, for having me. It's a great opportunity to present this research project, which is led by our PhD student, Yair Rokach, from the Hebrew University and Hadassah Medical Center. So our study is focusing on understanding the involvement of the gut microbiota in the pathogenesis and progression of HFpEF. Specifically, we're talking about the obesity phenotype of HFpEF. So we actually conducted several studies, including in mice and humans. First, we established mouse model with obesity-related HFpEF by high-fat diet and L-NAME, which is inducing both metabolic and hemodynamic stress in these mice. And this model has been shown to recapitulate several phenotypes of the human disease. Secondly, we also conducted a clinical trial recruiting patients with obese HFpEF phenotype. We collected samples, stool samples and blood samples, and compared this to two groups of control individuals with obesity, otherwise healthy individuals, and lean healthy subjects. So first, we examined the association between the gut microbiota and the presence of the disease in the different groups by sequencing of the microbiome. And we found significant segregation and differences in the composition of the microbial populations and communities, both in mice and humans. And this demonstrated the association between the gut microbial and the presence of the disease, representing also several pathogenic communities overrepresented in the HFpEF phenotype, both in humans and in mice. To provide evidence of causality and direct association between the gut microbiota and the manifestation of the disease, we performed two sets of experiments, including fecal microbial transplantation from humans to the mice after extensive antibiotic treatment to sterilize these microbiota of the mice, and generating humanized microbiota in these mice with the model. And we surprisingly saw a significant changes by echo when we look at the diastolic function in these mice. And we saw that there is more pronounced diastolic dysfunction occurred in mice after FMT from obese HFpEF patients compared to the control groups. And we look at the myocardial tissues. We also saw more pronounced inflammation and microvascular obliteration and endothelial dysfunction. We also performed experiments in a different group of mice called germ-free mice, which are sterile mice without microbiome. We did the same experiments of FMT from humans of the different groups, and we saw similar results indicating that transplantation of microbiota from patients resulted in more enhanced diastolic dysfunction by echo without affecting the systolic function and LV ejection fraction. And subsequently, we did also an analysis of the plasma and the heart tissues, first to look at the changes in systemic inflammatory markers. And we found increased pro-inflammatory cytokines, including IL-17, interferon alpha, IL-6, both in mice and humans after having these mice getting the microbiota from patients compared to the controls, and also in mice with the phenotype. Next, we wanted to provide mechanistic evidence of the changes at the blood level and the heart tissue level. And we performed RNA sequencing of the heart tissues in mice undergoing FMT from obese HFpEF versus control subjects. And we found briefly that FMT from obese patients resulted in a significant increase in hallmark hypertrophic fibrosis and inflammatory markers compared to the control groups. Then we did metabolomics using mass spec to look at metabolites both in the circulation and the hearts of these mice after FMT from different patient groups. And we found that the metabolic profile was significantly changed, and the groups are markedly separated between the different groups. And we can indicate that the profile is changed. And also we can point out several metabolites involved in amino acid metabolism, short-chain fatty acids, and energetic production has been changed and adversely affected in the HFpEF mice model with FMT from human patients. Lastly, we also did dynamic studies because we wanted to answer the question if FMT from healthy individuals can improve the diastolic function and if this process can be reversible. So we did a kind of double FMT to these mice from humans, and HFpEF followed by FMT from controls and vice versa. And we found that this process is reversible and we can improve the diastolic function by echo and also the inflammation at the myocardial level with FMT from healthy individuals. So if I conclude the main results of our project, that we demonstrate a causative link between gut microbial dysbiosis and HFpEF progression. Gut microbiota from obese HFpEF patients induce several inflammatory and pro-fibrotic signals implicated in worsening HFpEF phenotype in mice. And our study also suggests that susceptibility to HFpEF may be transmissible in obese individuals and can potentially be modified through gut microbiota manipulation as a therapeutic target.
Wow, what an elegant series of studies. So to kind of summarize, you can transplant bad microbiomes into another individual, in this case, a mouse. That makes some abnormal short-chain fatty acids and other things that get absorbed. They cause inflammation, and those inflammatory markers cause HFpEF. But if you put in a healthy microbiome, you can reverse all that. That is just fascinating.
Yeah.
So is this ready for, human to human transplantation at this point?
Not at this point. We are still doing the FMT to mice individuals. Actually, the clinical use of fecal microbial transplantation is only approved for specific diseases, mainly the Clostridium difficile infections, refractory ones.
Right.
Because of the issue of, the general transplantation of different microbes, the fear of transmitting specific pathogens that can be toxic and still not approved, and we need to probably go a long way to achieve this point. It's more attractive actually to find specific metabolites or specific communities that are favorable that can be transmitted and not a general stool transplantation. I think it's more elegant and more safe for the human utility.
I see. Well, just fascinating. One of the more fascinating things I've heard at this meeting. Well, thanks for joining us, and thanks to all of you for joining us here live from ESC 2024.
Thank you. Thank you so much for having me.